Hello everyone, this is Dr. Leili Hatami. Welcome back to another Physiology Lab session. Today I'm going to review the Physiology Lab Exam 1 study guide for Bio 249 Lab. It's important to know the equipment we use in the Physiology Lab. The first equipment that you can see the picture of it, it's called transfer pipette. So we have transfer pipette in this picture, which also known as disposable pipette. Which, if you remember, we disposed this item after we used it in the regular trash. Next, you will see this equipment, which is called serological pipette. So you have serological pipette in this picture. And then the next item is called bulb. If you remember, when we used the serological pipette, we used the bulb. And then the next item is called the microliter pipette. We use the microliter pipette to accurately measure and transfer the small volumes of solution. So make sure you know the name of this item, which is called the microliter pipette. To measure and transfer these small volumes of a solution, like 10 microliter, 20 microliter, 100 microliter, 500 microliter, and so on. Don't forget to take notes. Okay. And also it's important to know how to use it. The first stop, second stop, and ejector are the part of the microliter pipette, which is super important to remember how we use them. We are using the first stop to pick up the solution into the pipette tip from the container. So as you can see in this picture, when we are going to draw or pick up the solution into the pipette, we are using first stop. Okay, one more time. First stop to pick up the solution into the pipette tip from the container. And then when we are going to dispose the solution from the pipette tip into the new container, we are going to use the second stop. So second stop, when we are going to remove the solution from the pipette tip into the new container. So make sure you know the second stop and first stop. And then when you're using the microliter pipette, to remove the pipette tip without touching it, we use the ejector. Okay, next item in this view is called the graduated cylinder. So here we have the graduated cylinder, which we use this item to measure the volume of solution, like the large volume of the solution, like about 40 milliliter, 100 milliliter, and so on. So graduated cylinder to measure the large volume of the solution. Next item in view is called beaker. And then in this picture, you can see the flask. Here we have the test tube, which we use a lot in our physiology lab. So you should know or remember when we use the like test tube, we place them into the test tube collection bucket. So after using the test tube, so this item is called test tube. And after we use the test tube, we place them into the test tube collection bin or bucket. And then in the test tube, sometimes we had these liquid, the chemical liquid. To dispose the all liquids, we use the chemical waste container, which it was found near the sink in our lab. So the chemical waste container to dispose all the liquids inside of the test tubes. Okay, and then you can see this is parafilm. We use the parafilm to cover our test tubes or beakers for different reasons. And then now here you can see the test tube holder. Here we have the scale. So let me just write it down. Here we have the scale, which we measured the like amount of powder or something that we need to weight them. So we use the scale in our lab. And look at this huge device, which is called spectrophotometer. So I'm sure you remember this spectrophotometer, which we use this to measure the absorbance and transmittance of a solution. So you have to know the function of this device for measuring the absorbance and also the transmittance of a solution. We use this device. Next in view, we have the fume hood. Then you can see these equipments we use for testing the reflexes. The picture in left side, it's called the reflex hammer. As you can see in this picture, the physician is testing the reflex, like the knee-jerk reflex, by using the reflex hammer. And then the picture in the right side is called reflex light, which we use the reflex light to assess the pupillary light reflex. And next in this picture, you can see the lancet. Make sure you know the scientific method steps. Scientific method is used to help us learn about the world around us. So the first step is through observation and research, and then second, there are gonna be a question, and the third one is the step involve a lot of background knowledge and also if then statement, which is called the hypothesis or the scientific guess. And then the next step is to design the experiment to test our hypothesis. And after designing the experiment and running the experiment, we are going to collect the data. Then this step that we are going to analyze the data through graphing is called the data analysis. And next we are going to take the conclusion, and the last step is the scientific communication, which can be through like publication of the article or presentation of the result. So one more time, make sure you know the first step of the scientific method is the research and observation, and then the hypothesis is a scientific guess, which during this step you are going to make a statement as if then a statement, and after making the hypothesis, we are going to test our hypothesis through the designing the experiment. Collecting data, it's gonna happen through the experiment, and then when we are going to analyze the data through graphing or something like that, we call this as data analysis. And then the last step of the scientific method is called the scientific communication. We learned how to design a good experiment, and through that process we learned there are some experimental variables. You should be able to identify the independent variable, dependent variable, and also standardized variable. Make sure to know the definition of each of these variables, so it's going to be so easy to find them in each experimental study. For independent variable, make sure you know this is a variable that the researcher is going to test that variable, and also is able to change or control it. So the variable that is going to be tested is called independent variable, or the researcher is able to change or control it, it's called independent variable. Dependent variable is the variable that is going to be measured or observed, and the scientist or researcher cannot change this variable. They only can observe it or measure it. So these are the keywords for dependent variable. And a standardized variable is the variables that are going to be the same during the experiment. Okay, so let's read this example and practice. In an experiment examining the effect of the dietary iron intake on blood iron levels, you recruit 50 participants. They are divided into two groups. One consumers iron one consumes iron fortified breakfast cereal, and the others consumes cereal without added iron. And now you should be able to find the independent variable, dependent variable, and standardized variable. Okay, let's review the independent variable one more time. The independent variable is the variable that's going to be tested. So it can be tested. The researcher can control it and can change it. So here we have the type of breakfast cereal which the researcher is willing to test it. So then it can control it, it can change it. So for example, the iron fortified breakfast cereal versus non-iron fortified breakfast cereal. So then the independent variable would be the type of breakfast cereal. For example, non-iron fortified versus iron fortified. As we saw in our two groups, we had the iron fortified breakfast cereal versus without added iron. And what about the dependent variable? Dependent variable is the variable that we are able to measure that. Okay, so we're gonna observe it or measure it. What we are observing? We are observing the blood iron levels. So that would be our are dependent variable levels. For the standardized variable, it actually include factors such as the amount of cereal consumed, the duration of the study, the participants' age and gender, their overall diet, the physical activity levels, and any other factors that could influence blood iron levels. And we have to make sure to have them in the same during our study. Okay, and then you should know what type of graph would best show the data you collected. So in our physiology lab, we learn about these three type of graphing. We have bar graph, line graph, and scatter plot. So the first thing you have to know is how to define them and find the keyword for each of these graphs. The bar graph is used to compare the discrete numbers, typically averages. The line graph is used to show a pattern for an individual over time. So that is our key. So when we are going to like compare two categories over time, we are using the line graph. And then when we are going to show the correlation or the relationship between two variables, we are going to use the scatter plot. So the correlation or relation is the keywords that help us to find the scatter plot is the best graph that can show our data that we collected. Okay, and then here you have a question, less practice. You want to determine if you and your mom have a different blood albumin level. You test your blood once a day for a month. You test your mom's blood once a day for a month. What does it mean? Over time, right? What type of graph should you make to show your data? So as we discussed, when we are going to compare two different categories over time, so we are going to use the line graph. So the best graph for this experiment would be line graph. And we have another example. You're researching the link between weekly exercise hours and weight loss for individual on a specific diet plan. Each participant's data includes their weekly exercise hours and weight loss over 3 months. So the link, it means like the relation, the correlation. So it's important to know that the scatter plot is the best for this experiment. Okay, and also during our physiology lab, we use different reagents and indicators. We use glucose reagent, iodine test or reagent, Benedict's reagent, biorate reagent, and litmus indicator. It's super important to know each reagent is indicating for which factor or molecule, and then what's going to be the positive or negative test for each of these indicator or reagent. The glucose reagent, the first one, which is super obvious, the glucose reagent is going to test the glucose for testing glucose. The iodine test, it's gonna be used for starch. Benedict's reagent is the reagent to test for, it's gonna be test for simple sugar, like monosaccharide, like again glucose. Biorate reagent is gonna be used for testing proteins. And then litmus indicator, it's gonna be used for pH. So it's a pH indicator. If you remember, we used the pH indicator, the litmus indicator in our fat digestion lab. Okay, so now let's go over each of these to know more. The glucose reagent is the reagent that we can test for glucose. So as you can see and remember, the glucose reagent itself is like amber color or orange color. So when we have glucose inside of our solution, then the glucose reagent color would be changed to red. So without glucose, just glucose reagent and water, we have the orange color or amber color of our glucose reagent. But when the color gonna be changed to the red, it means there is glucose present in our solution. So glucose reagent will show which color. When glucose is present, it's gonna show or the red color. When it's amber color or orange color, it means no glucose. So orange color, no glucose. Red color, yes, glucose. Next, we have Benedict's reagent. The Benedict reagent is another reagent for small or simple sugars like monosaccharide, disaccharide. So as you can see, the Benedict reagent itself color is blue, but when we have the simple sugar like monosaccharide, disaccharide, then the solution color after adding the Benedict reagent gonna be changed to red or orange or yellow or green. So as you can see in this picture, we have 5 test tubes which we have like we added the Benedicts. So Benedict reagent to each of these. And you saw that the first one on the right side, it stays blue, which means no color change. So that our test is negative. It means that there is no like monosaccharide or like glucose inside of no glucose inside of our solution. But when we got the green color, yellow color, orange color, red, or ROIG, it means the test is positive. It means we should have like monosaccharide, like glucose inside of our solution. Next, we use the iodine test in our physiology lab. The iodine itself is called, it's like a yellow color or again amber color. And the iodine gonna be used for starch. When iodine is absent, the after adding the iodine, so the color is still yellow. But if we can see the changing of the color from yellow to blue or purple or dark blue, it means this starch is present and our test is positive. So iodine is a test for starch. One more time. If we have starch present in our solution, then that would be blue color or black or dark. Next reagent is biuret reagent, which is an indicator for protein. You can see if there in the solution there is no protein, the solution color remains blue. So no color change. It means our test is negative. It means there is no protein inside. So no protein inside, our binary reagent stays blue. But if we have protein inside of the solution, then we can see the purple color. So the purple color shows there is protein in our solution. And also we use the litmus indicator, which is a pH indicator. We use this during our fat digestion lab. Litmus will turn the color to dark blue. When the solution is basic, litmus will turn the color to the red or pink when the solution is acidic. So as you can see in this picture, we can get these three color with the litmus. The dark blue, it shows the pH is alkaline above 7. pH, it's going to be above 7, or is basic or alkaline. If we got the pink or red color, it's showing the pH is less than 7. It means the solution is acidic. And if we get the violet color, or like a light purple or lavender color, it means our pH is neutral. So in this picture, you can see two kind of the first one for sure is purple. What does it mean? It means the pH should be around 7, or we have neutral pH, right? And then look at this one, is like a pink. So it means the pH is less than 7, it's acidic. And then this one is also is acidic, but as you can see, this one is more like to red than this one. So it means the acidity of this test tube is more than this. Let me just name these as number 1, 2, 3, and 4. So it means the test tube number 3 solution is more acidic than number 2. And then the last one, it's kind of getting to the pink from the lavender. It's going toward the acidic, but still it can be like neutral toward the, so let me write it down, neutral toward acidic. Okay, so when we use the pH indicator during our fat digestion lab, as you remember, after digestion or breaking down the triglyceride, we got the fatty acids, and that was the reason our digested fat solution, or the solution it has digested fat, it's getting more acidic. So if you want to compare this and to guess which one has digested fat, we can say this test tube number 3, it has more digested fat than this one. Okay, so then we have to know the spectrophotometer and some of the terms like transmittance and blank solution, and also the absorbance. So the spectrophotometer, which we discussed about this device before, is a device that we are going to measure the absorbance and transmittance of a solution. But what does it mean, absorbance? Absorbance is the amount of light that the sample is going to block it. And transmittance is the amount of light that passes through the sample. So one more time, absorbance, as the name of this term is descriptive, is going to be absorbed through the sample. So we are going to pass the light through the sample, but the amount of light that the sample is going to block it is called absorbance. But the amount of the light that it's going to be passed through the sample is called transmittance. And also, if you remember, we use the blank solution to calibrate our spectrophotometer. A simple definition of the blank solution is a solution which contains everything except the molecule or item you are testing for. And then the first thing that we did, we place the blank solution in our spectrophotometer, and then we adjusted the absorbance of the blank solution to the 0 to calibrate the spectrophotometer. So it's super important to know. And also we had some activity in our lab assignment we set the absorbance of the blank solution to the 0 to calibrate the spectrophotometer. Okay. Next, you have to know the concept about the concentration and absorbance. One of the way that we could measure the concentration of our unknown solution, it was through the Beer's law. In the Beer's law, we have to have the concentration of our standard solution, and then we measure the absorbance of that standard solution, and also we could measure the absorbance of our unknown solution, but the one that it was missing, it was a concentration of the unknown solution. During our experiment, we learned that as concentration increases, the absorbance also increases. And the concentration when decreases, the absorbance also decreases. There is a linear relationship between the concentration of a solute in a solution and the absorbance of light by that solution, which is makes sense because more molecules they're gonna be there to absorb the light. So when the concentration of substance increases in a solution, there are more molecules of that substance present in the solution, so they're gonna absorb more light. Okay, and also in this one, we are going to practice how to use the Beer's law to count the unknown solution concentration. In this example, we have the absorbance of our unknown glucose solution 0.080. So let's write it down. We have the absorbance of, we are going to show the absorbance by letter A, our unknown solution X, which is 0.080. And we know there is no unit for absorbance. And then we have the absorbance for our standard glucose solution as 0.0110. So then would be the absorbance of our standard solution that is 0.0110. Oh, let me fix my zeros. Okay. But what do we have here as well? 1 milligram per deciliter is the concentration of our standard solution. So we have another number. So let's put it here. We have the CS, which is the concentration of our standard solution, is 1 milligram per deciliter. Please do not forget to add the units. And now is asking to calculate the concentration of unknown glucose solution. So we are using the Beer's law. For the Beer's law, we have the CS, which is the concentration of our standard solution, divided by AS, which is the absorbance of our standard solution, equals the Cx, the concentration of our unknown solution, divided by AX, which is the absorbance of our unknown solution. So just simply put all of the number in their place. For CS, we have 1. For AS, we have 0.010. Then for CX, just place 6 here. For AX, we have 0.080. Okay. So then just do a simple math and you should get this number, which our CX would be 8. Don't forget the unit, milligram per deciliter. And also there is another way that we can calculate the unknown solution concentration, which is called a standard curve. For graphing the standard curve, we need some numbers. So through the MS Excel, we can draw the standard curve, and then it's gonna give us the equation. The straight line on standard curve is the equation for this curve is y equal mx plus b, which y is the absorbance of our unknown solution, m is the slope, the rise and run, which the by drawing this standard curve is gonna give us this number. So this number is given, and then the x is concentration, the b is y-intercept, and also we can get this number also through drawing this standard curve. So then we have to put all of the number in this equation. So again, y is the absorbance of our unknown solution. In this case, we have absorbance of our unknown solution as this number. So we are going to put here 0.615 equals m, which our m is here given. So we have 0.422 multiply x. So we do not have the x, which we have to calculate the x, plus then here we have the b, which the b is also given through the standard curve, and we are going to place this number here. Then you have to do the simple math to find the x. So hopefully you found this number. So the concentration of our unknown solution would be 1.444 milligram per milliliter, because we have this unit here. Next concept which we have to know is the definition of the semi-permeable membrane. The semi-permeable membrane is the membrane which it's gonna let some solute or substances passively pass through the membrane. So it means if a solute is permeable to membrane, then it has the ability to passively diffuse through the membrane. So for example, in this picture, you can see the membrane is semi-permeable membrane and is letting the water molecule to passively diffuse through the membrane. But salt is not permeable to the membrane, so then we need to actively or through using some equipment, using some energy to move the salt around. So again, one more time, make sure you know that if a solute is permeable to the membrane, then it has the ability to passively diffuse through the membrane. If a solute is impermeable to the membrane, then it cannot diffuse passively through the membrane. Okay, next concept is osmosis and osmotic rate. The osmosis is movement of the water from a higher concentration of water to the lower concentration of water, or the movement of water molecule across a semi-permeable membrane from our area of the lower solute concentration to an area of the higher solute concentration. So let's look at this image, and in this image you can see here we have a semi-permeable membrane in between, and then there are two different solutions with different tonicities in the either side of this membrane. So as you can see, if you compare the inside of the cell solution to the outside of the cell solution, you can see the outside we have less solute. We call this solution as a hypotonic solution, which means it has less solute but more water in comparison with the solution we have inside of the cell. And we learned that the osmosis is the movement of molecules, the water molecules, from more water to less water, or from less solute to more solute. So then, as you can see here, we have the movement of water from the solution, from the hypotonic solution to the cell, or from the solution with more water, less solute, to the solution with less water, more solute. And then the other example, you can see the same amount of solute inside and outside of the cell, which means we have the isotonic solution, so same amount of solute, same amount of water. So then the net, or the movement of water between these two, is zero. So the net movement is zero. And then we have the hypertonic solution, which, as you can see here, we have the more solute in the extracellular space. So we have more solute in this solution, which means we have less water in comparison with the inside of the cell. So it means literally our solution is a hypertonic solution. And let's follow the movement of water. Then water is going to move from less solute to more solute, or water is going to move from higher concentration of water to lower concentration of water. Okay, and let's practice one more time. The osmosis causes water molecules to move out of the area with which amount of solute concentration. So osmosis causes the water molecule is moving out of the area with the less amount of or low solute concentration, from low solute concentration to more solute concentration. Or osmosis causes water molecules to be pulled towards the area with, again, high or more solute concentration. And also there is another rule which is related for osmotic rate. The more the gradient we have, the faster rate. So you can see here the steeper concentration gradient is gonna let we have the faster rate of the osmosis. The less the gradient, the slower rate will be, or the less steep concentration gradient, and then we have slow rate of diffusion. What is the gradient? Gradient is the difference between concentration of two different solutions that we have. So for example, if we have the concentration of our solute, which is like 8%, and another solution which is 1%, 8 minus 1, the 7, it would be the concentration gradient for these two solutions. And when we have the more gradient, then we are going to have the faster osmotic rate. When we have the less gradient, we would have the slower rate of diffusion or osmosis. Okay, let's practice through this graph. So look at this graph, and it's asking you what is the slowest osmotic rate. Okay, so in our experiment, it seems we had 4 dialysis bag, or 4 artificial cells. Bag A, it's showing with the blue color, which you can see in the graph. The x-axis is showing us the time per minute. So we had 0 time to 60 minutes, 0 to 60 minutes. And then our y-axis is showing the weight of the bag, or our artificial cell. So the bag A started at 10 gram, then after 1 hour or 60 minutes, we see the bag A lost weight. What does it mean? The losing weight, it means that our cell lost weight. It means that the water went out of the cell. So do you remember when it's happening? When we are going to place our cell inside of a hypertonic solution, that would happen. So when we are going to place our cell to the hypertonic solution, then our cell is going to lose the weight. So that's happened for our bag A, which is blue color. Then the orange color, you can see the bag B. At the minute of 0, it was 10 gram. And then at the minute of 60 also, it is 10 grams. So there is no changes at the weight. What does it mean? It means that bag A, it seems we place the bag A inside of the isotonic solution because it was not any change in their weight. And then look at the bag C, which is the gray color. It means the bag C also gained weight. So it's gained weight exactly like our bag D. These two gained weight. It means that we placed them inside of the hypotonic solution as the water went inside, and that was the reason our bag weight has been increased. But if you look at this, it's asking the slowest osmotic rate. So we actually should find the slowest steep. So which of these lines it's showing us the slowest steep, which is the bag C, the gray color, because bag orange or bag B doesn't show any osmotic rate. The osmotic rate is 0, but when it's asking the slowest osmotic rate, that should be the one it has low steep, which is our bag C. Okay. Again, the concept of the hypertonic, isotonic, and hypotonic solution is super important and very useful in the clinic. The hypertonic is the solution. It has more solute, less water. The isotonic solution has the same solute and water in comparison with the inside of the cell. Hypotonic solution, it has less solute and more water. Okay. So for our body, the isotonic solution, which is the safest solution for IV, is 0.9% normal saline or NaCl. That is our isotonic solution. Any number above 0.9 consider as the hypertonic. For example, 1.3% NaCl, 3% NaCl. So these consider as hypertonic, and any number below this number consider as hypotonic. So for example, 0.5% NaCl, even 0.8% NaCl, that would be considered as hypotonic. So the best one, it considered as isotonic, is 0.9%. Okay, so if you place your cells inside of any hypotonic solution, as I mentioned, less than 0.9%, then you can see your cell is receiving the water, or the water is actually moving inside of the cell, because the outside or the hypotonic solution has more water than solute. So then water moving from higher concentration of water to lower concentration of water, or from the solution which has less solute to the solution it has more solute. And if we place our cells into the hypertonic solution, it means any number above 0.9%. So then the cell is going to lose the water because the water again through osmosis is moving out because the water is going to move from higher concentration, which in this scenario, in the hypertonic solution, we have more solute outside, less water outside. Then water moving out from inside of the cell to the outside of the cell. Okay, so let's practice one more time. The osmotic rate, hypertonic, isotonic, hypotonic solution. We have a case, dialysis bag A contains 5% NaCl. We have another dialysis bag B, which contains 4% NaCl. And then the dialysis bag C contains 1% NaCl. So we have the A, 5%. The dialysis bag B, it has 4%. And then our last one, which is the dialysis bag C, it has 1% NaCl. And we place them inside of the solution, which has 2% NaCl. Okay. And now is asking which bag will exhibit the slowest osmotic rate. So one more time, just remember that. This rule, this pattern, the more the gradient, then the faster osmotic rate will be. The less the gradient, then the slower osmotic rate will be. So we have to find the concentration gradient between these solutions. So in our dialysis bag A, between these two solutions, we have 5 minus 2, 3. It would be the concentration gradient for our bag A. Then for the B, we have 4 minus 2, which 2 is the concentration gradient between these two solutions. And last one, we have 2 minus 1. 1 would be the concentration gradient for between these two solutions. Okay, so based on our rule, which has the less gradient, that would have the slow rate. Which one has the less? Between 3, 2, and 1, the like dialysis bag C, it has the less gradient, so then it would have the slowest osmotic rate between these are three like cells or bags. Another one, dialysis bag A contains 3% NaCl. Dialysis bag B, it has 2% NaCl, and dialysis bag C contains 1% NaCl. And we placed all of them in a 2% NaCl solution. Let's have one beaker which has 2% NaCl, and then we have 3 bag or cell, A, B, and C, 3%, which means like the amount of solute, that would be 3, and amount of solvent, that would be 97. And then the other one is 2% NaCl, 2% for solute, 98% for solvent, and then C is 1%. And then our beaker has 2% NaCl solution, which bags were placed in a hypertonic solution. The easiest thing that we can do is that easily we can cross out the bag B because it has the same solute concentration in comparison with the solution inside of the beaker. So it means the B is play for the bag B, the beaker is isotonic. For the bag B, the outside solution is isotonic. So isotonic for bag B. How about for bag C? So it means in our bag C, we have 1%, which means less solute in comparison with the solution we have outside. So it means our solution, outside solution, is more concentrated than inside solution. So that would be the hypertonic. So it means our beaker or outside solution is hypertonic in comparison with the bag C. And if you look at the concentration of the solute inside of the cell, it seems we place the bag A inside of the hypotonic solution. It's a little bit confusing, but let me to do this. So actually it means just know that the outside solution is isotonic for bag B, right? Which is obvious. And then we are going to compare the outside solution tonicity with the bag C. So the outside solution, it has more solute. It has more solute than bag C. What does it mean? It means that the outside solution is hypertonic to bag C. And if we do the same for our bag A, we can say the outside solution has less solute, more water than bag A. So then it means the outside solution is hypotonic for bag A. Okay, so the next concept is how to normalize the weight. So if you remember in one of our lab, we actually needed to normalize the weight. To normalize the weight, because each dialysis bag begins at the different weight in grams. We could not compare the data in grams because the amount of change may be affected by the initial size of the cell. So then we needed to normalize the data in order to compare the bags to each other. So then normalizing data means that we transform our data so that they are both at the same scale of measurement. We are going to transform from gram to percentage. Then we can say now our weight is normalized, and then we accurately can compare our groups to each other. So then we have a formula for normalizing the weight. Please make sure to memorize this formula, which the normalized weight per percent, it means weight in grams at the time of 0 divided by weight in grams at the time of, not 0, sorry, one more time. Weight in grams at the time of X, which can be 15, 20, any time that we want to normalize it, divide by weight in grams at the time of 0, multiply 100, and then percent is going to be counted as our unit. So let's practice with this example. You place a dialysis bag filled with 5% sucrose solution in distilled water, and then the initial weight was 5.3 gram, and the final weight after 15 minutes was 7.2 gram. And now we need to normalize the weight because of the reason I mentioned. So then what do we need? We actually want to calculate the normalized weight. So the normalized weight per percent, then weight in grams at the time of X, our x, that would be 15 minutes. So what is the weight of our like solution or bag in the time of 15, which is 5.2 divide by weight in grams at time of 0. The initial weight, which gonna be the time of 0, the weight was 5.3 multiplied by 100. And then just a simple math, and you should get this number. The number should be like average 5.81, which we can round it to 136%. Okay, so it means we normalized the weight or transformed the weight from gram to the percentage. Okay. Next concept is solubility, hydrophilic, hydrophobic, polar, and non-polar molecules. If you remember, we have a general pattern which it says the hydrophobic material dissolves in hydrophobic material, or hydrophilic material dissolves in hydrophilic materials. Hydrophobic materials like oil dissolves in hydrophobic materials. Hydrophilic materials like water dissolved in hydrophilic materials, which like the material which they like water as well. Okay, and then we have example is asking you mix 2 ml of water. Okay, what kind of molecule is that? Water is polar molecule. Any molecule they love water and can be dissolved in water, these are the hydrophilic molecules. And then we mix this water with the 2 milliliter of olive oil. The olive oil or any vegetable oil or any oil, these are the non-polar molecules, which also they are considered as the hydrophobic molecules, or the molecules they hate water and they're not going to be dissolved in water. Okay, and then you added a dye and then shake the mixture. After shaking, you observe that water turns colored. So if we add the water and oil, we know the water, it remains on the bottom, and then oil gonna be found here on top. Okay, so we have the oil here on top. But after adding the dye, we saw that dye mixed in the water. So now our water is colored. What does it mean? It means the dye should be loving water. It means hydrophilic. Or the dye would be polar molecule. So one more time, any molecule that they can be dissolved in water, this is considered as hydrophilic or polar molecule. And any like molecule that cannot be dissolved in water, they are called hydrophobic, which they are non-polar molecule. And also we learned the concept of micelles. Micelles are the structure composed of the amphipathic molecules, which the amphipathic molecules are the molecules which they have both hydrophilic and hydrophobic regions. So as you can see here, this circular structure is called micelles, which is important to know that micelles gonna carry the oil, which is the hydrophobic molecule, so like oil, safely through the water. So we have water outside of the micelle and oil inside of the water micelle. So the micelles carry hydrophobic molecules safely through water. Next, we should know the amylase, which the amylase is an enzyme, is the enzyme to digest the carbohydrate. So amylase enzyme is using for digestion of carbohydrate. We have salivary amylase, which is going to be found in saliva. And also we have the pancreatic amylase, which the pancreatic amylase is going to be released by our pancreas, and also the pancreatic amylase is going to be found in pancreatic juice and then inside of the small intestine. So the question like this, you should answer the amylase is secreted in saliva and also in pancreatic juice. Do we have amylase in gastric juice? No, there is no amylase in gastric juice. So we have salivary amylase and pancreatic amylase. It's going to be found in saliva and pancreatic juice. So as I mentioned, the amylase is going to be used for carbohydrate digestion. So by using the amylase, we can digest the carbohydrate like from polysaccharide to the monosaccharide, from the larger molecule of sugar or carbohydrate to very like simple sugar molecule. The question asking if you increase the amount of amylase activity, what would happen? So if we have the increasing the amylase activity, it means that we are going to digest or break down more polysaccharide carbohydrate to monosaccharide. And then what's going to happen for the amount of monosaccharide? So obviously, if we are going to increase the amylase activity, then we would have more amount of monosaccharide. So the amount of monosaccharide will increase. Okay, because we know the amylase is the enzyme breaking down polysaccharide into the monosaccharide. More amylase activity, we would have more monosaccharide. Less amylase activity, we would have less monosaccharide. Okay, and then these two terms are also important, the polymer of carbohydrates and monomer of the carbohydrates, which the polymer of carbohydrates are polysaccharide and monomer of carbohydrates are monosaccharide. So we have polysaccharide for polymer of carbohydrates, and carbohydrate monomers would be monosaccharide. Okay, speaking about the fat, lipids, so they can differ in their shape. We have saturated fatty acid and unsaturated fatty acid. If there are single bonds between these molecules, we call them as saturated fatty acid, which they remain solid at the room temperature, found in animals like meat, milk, cheese, eggs, or in plants like coconut oil, and then they actually can raise cholesterol level. But unsaturated fatty acid, they have double bond present, and if you look at their structure, you can see a kink in their like molecule. So these are the keywords: finding the kink in their structure or double bonds present. It's showing the unsaturated fatty acid like olive oil and sunflower oil, and if they have all single bonds would be saturated fatty acid like coconut oil. Okay, next enzyme is pepsin, which the pepsin is the enzyme used for breaking down the protein and gonna be found in gastric juice. So pepsin is secreted in the gastric juice. Protein and protease. Protease is another enzyme for breaking down protein, which gonna be secreted through the pancreas. But the question asking, if you increase the amount of protease activity, then what gonna happen for the amount of protein? So as you can see in this picture, the protease is the enzyme that can facilitate breaking down of protein to amino acid. So it means more protease activity, we would have more protein breakdown, right? So when we are going to break down protein, we are going to get more amino acid. So if we increase the amount of or the protease activity, so then we would have less protein. So the amount of protein decrease, but instead the amount of amino acid would be increased. Okay, another concept related to the protein and protease. Proteases will help break down the peptide or peptide bonds that we can see between the amino acids, and then hydrolysis is the reaction which breaking down the polymers into the monomers. So make sure you know these two terms, the peptide bonds which the proteases can help to break them down, and also the hydrolysis is the process which also can break down the polymers into the monomers. Okay, so then we had the experiment for protein digestion, which we are going to practice how to find the negative control in our protein digestion experiment. In the protein digestion experiment, you added the biuret, which if you remember, the biuret was the indicator for protein, and albumin, which is our protein. So we have the biuret as indicator or reagent for protein. Albumin is our protein, okay? And then you place all the tubes at 37 degrees centigrade, which is the optimal like temperature for our enzyme. And what else did you add to the negative control? So negative control is the group that we are sure that nothing gonna happen because we did not add the enzyme for protein digestion to happen. So it means our negative control, it has the biuret, the indicator, the albumin, and then we added the water also to make sure that we have a negative control. So what else did you add to the negative control? It was water. Next, let's practice for optimal environment. We learned that each enzyme, they have an optimal environment. Their pH should be in optimal environment, optimal pH. Their temperature should be in optimal temperature, which at that optimal environment, they can have their optimal enzymatic activity. You're studying pepsin. Speaking about the pepsin, just remember pepsin gonna be found in gastric juice in our stomach. Pepsin is the enzyme to break down the protein. Speaking about the stomach, just remember that in the stomach we have the pH of like 1, 2, 3, like an acidic pH. Okay, now let's read the rest. The control experiment will have a pH that is what? Which one? So obviously pepsin, because it's going to be found in gastric juice in our stomach, so we expect to have the optimal pH as the acidic. So the pH, it would be acidic, and the optimal temperature would be body temperature. So what is the optimal pH for pepsin? Again, think about the environment of your stomach. Our stomach is acidic, then the optimal pH for pepsin is also the acidic pH. What is the optimal temperature for pepsin? What is the environment of our stomach? It's 37 degrees centigrade, which is the temperature in our stomach. That would be the optimal temperature for our pepsin. Another practice for optimal environment and optimal enzymatic activity. You are testing the enzyme activity of gastric lipase. Again, speaking about the stomach. So lipase is the enzyme to break down the fat or lipid, and gastric, it means this enzyme should work in the stomach. Okay, so then the enzyme would have a higher enzymatic activity at which pH? slightly acidic, basic, or neutral. So obviously, think about the environment of the stomach as this enzyme it's working inside of the stomach. It should have in the acidic environment, it should have the higher enzymatic activity. Another practice for optimal environment and optimal enzymatic activity. Identify the optimal pH for enzyme 2. In this graph, you can see three different enzymes. For enzyme 2 is actually asking which one is the optimal enzymatic like temperature. Okay, so then look at this. In this graph, you can see here we have temperature, and then here we have the pH. Enzyme 2 is showing with the blue color, and this is the point which we have the highest activity of our enzyme. So if you go down, then you can find the optimal pH for our enzyme 2, that would be 6. Identify the optimal temperature for enzyme A. Again, in this graph, you can see our y-axis is showing the activity for enzyme, which the enzyme A, the highest activity would be here. So then the optimal pH, the optimal temperature for enzyme A would be 37, and also the optimal pH for enzyme A would be 7. Fat and lipase. Lipase is the enzyme which it can break down the lipid. And it's super important to know the lipase can be found in saliva. Lipase enzyme can be found in our stomach. And also lipase can be found in our pancreatic juice. Okay. If you increase the amount of lipase activity, what would happen for the amount of free fatty acids? So next thing that you have to know is that when we are going to break down the lipid like the triglyceride by using the lipase, then our product would be one molecule of monoglyceride and two free fatty acids. So it means by breaking down the triglyceride, we are going to get fatty acids, which this fatty acid is going to make our solution more acidic, right? We learned that before. And then it says when we are going to increase the amount of lipase activity, obviously we are going to get more free fatty acid. So the amount of free fatty acid will increase, and also we can say that our solution would be more acidic because of the increasing the amount of free fatty acid. Okay, so then another question related to the fat digestion, but this one is asking about the positive control. In the lipase and bile experiment, you added cream and litmus to all the tubes. What else did you add to the positive control? Just remember that the positive control is an experiment. The positive control of an experiment is a setup that is known to produce a positive effect. We expect to see the positive effect or positive outcome. So for our lipid digestion, we have to make sure we have all the ingredients needed to break down the lipid, like enzyme and also bile. So in asking what we have to have in our positive control, let's see, do we have the enzyme? Do we have the bile? You added cream, which is our lipid. Okay, and then we added the litmus, which is our indicator. What else did you add to the positive control? That should be the bile and also the enzyme, lipase enzyme. So just remember the fat digestion lab which we did. We set up our test tubes to the negative control, the positive control. Our positive control, it has the lipid, lipase, which is an enzyme, and bile to break down or physically or mechanically break down a lipid to increase the surface area, and the lipase enzyme can easily break them down. Then our negative control, we do not have enzyme, do not have bile, only lipid, which we are sure that nothing gonna happen in the negative control. Okay, speaking about the lipase, so let me introduce you the gastric lipase, which the gastric lipase is the enzyme gonna be found in our stomach. And then here is asking, you are testing the enzyme activity of gastric lipase. This enzyme will chemically digest the fat and it will change the pH of the stomach by making it more acidic, neutral, or basic. What do we expect when we are going to break down the lipid or triglyceride by using the lipase to the fatty acid and monoglyceride? Literally, we are going to increase the acidity of our solution. So it will change the pH of our stomach by making it more acidic, because now we have more fatty acid by breaking down the lipid. Bile. Bile is the product of the liver, which it can mechanically break down the fat and to increase the surface area. So make sure you know the bile increasing the mechanical digestion, and then it's the lipase rate will increase when bile is present. Is it correct or is not correct? So the lipase, which is the enzyme, obviously it depends to the action of the bile. When the bile is active and it's kind of actively breaking down the fat, increasing the surface area, we expect the lipase and enzyme rate would be increased as well. So lipase enzyme rate will increase when bile is present, which is true. Okay. Okay, so next concept is regarding the goldfish insulin shock experiment, and which we have to find which one is the control experiment. During the goldfish insulin shock experiment, we had the goldfish in only water, and goldfish in insulin, and goldfish in 5% glucose. So the control experiment, the purpose of the control experiment is to provide a baseline for comparison. So then allowing the researchers to determine the specific effects of the insulin on the goldfish glucose levels, behavior, or other parameters. So then to create the control group, we are going to remove the factor that we are going to test it, like insulin or 5% glucose, which means the water, the group which we only have water, that would be our control group. These are control group which we are going to compare the other groups which are control group. Okay. So, and also we did the glucose, the blood glucose regulation experiment through the goldfish. And now it's asking, when we are going to place our goldfish in insulin, what's gonna happen for their blood glucose level? Obviously, their blood glucose level would change. So, because the goldfish can absorb the insulin, and we know that insulin is going to decrease their plasma glucose. So then the insulin is gonna impact their plasma glucose to decrease. So then which cells in their body can detect these changes? This receptor or the sensor would be found in their pancreas, which the cells are so sensitive regarding the decreasing of the plasma glucose is going to be the alpha cells. So the sensor would be the alpha cells found in the pancreas. Okay, so these alpha cells of the pancreas, they can detect the plasma glucose level had been changed, and also they itself are controller because they are gonna mm, produce and release the hormone to regulate the blood glucose level. So then identify the controller. The controller also would be the alpha cells of pancreas. So which they gonna produce the glucagon hormone and release them into the blood, and the glucagon through the blood circulation it goes to the target cells or target organs, which are the target cells or target organs for glucagon would be liver cells or hepatocytes, and also would be the muscle, also would be the fat tissue or adipose tissue, and also would be all the cells or other cells in our body. Okay, and the next example, when we place the fish in 5% glucose, what happened? So when we added the fish inside of the 5% glucose, they started to absorb the glucose inside their blood, right? So it means that their blood glucose level has been increased, exactly like we had a sweet meal. So then we can see increasing of where is it? Increase in their like sugar level or glucose, the blood glucose level. Okay. So that is our change. What's gonna be the receptors or their sensors? The receptors at this time increasing the plasma glucose, the receptors or sensors would be the beta cells of the pancreas. And also beta cells of the pancreas is gonna be their controller as well. So we have sensor or receptor, the beta cells of the pancreas, and also they're gonna be controller because they can like regulate the blood glucose level from high to low. So then this insulin through the blood circulation, these beta cells gonna produce the insulin, and the insulin through the blood circulation, it goes to these target cells, which is liver, muscle tissue, adipose, and the other cells. Okay, so then let me just go back to the glucagon. I'm going to correct my mistake that I had. So for glucagon, the target would be only liver or hepatocytes, okay? So we have the target for glucagon, liver, but for the insulin, we have more target, liver, muscle, adipose, and other cells. So we have the target for the insulin as liver, muscle, fat tissue, and also the all body cells. They have receptor for insulin. Where is insulin produced? Obviously in pancreas by beta cells. Okay, another example for blood glucose regulation, you ate a chocolate bar, your body is regulating this change in blood sugar. Identify the controller for this feedback loop. So when we ate the chocolate, so it means that we change our blood glucose level. So it means we have now experiencing increasing of the glucose level in our blood. Okay, so then what would be our receptor? The increasing the glucose going to be noticed by beta cells of the pancreas, and also they are the controller. So beta cells of the pancreas, they are going to release the insulin to the blood, and the insulin is going to liver, is going to fat tissue, it's going to muscle tissue, and also the all body cells. trying to lower the blood glucose level. Another example says during 6 hours of fasting, blood glucose level will. When you're fasting, it means that your blood glucose level decreases, right? Let's practice. When we have decreasing in blood glucose level, what would be our sensor or receptor? The alpha cells of the pancreas. What would be our controller? Again, the alpha cells of the pancreas, which they're gonna produce and release the glucagon into the blood, and then the glucagon, it goes to the liver, which is the target to increase the blood glucose level. Okay, we're almost done. Let's focus on the stretch reflex. The stretch reflex was one of the activity we did in our last lab. So you should know the stretch reflex components, which is receptor, afferent pathway, control center, the efferent pathway, and effector. At first, we need the stimulus by tapping through using a reflex hammer on a tendon to quickly stretch the muscle lengths. We are going to kind of letting the receptor knows something is happening. So the receptor for stretch reflex is going to be found inside of the muscle, within the muscle, which is called stretch receptor. So the receptor for stretch reflex is called stretch receptor, or also known as muscle spindle receptor, which these receptors are so sensitive about the muscle lengths. By tapping on the tendon and quickly stretching the length of muscle, these receptors can send the signals through the afferent pathway, which the afferent pathway is the sensory neurons. They're gonna let our control center know about these changes, which the control center of all the stretch reflexes are spinal cord. And then the afferent pathway, which would be the motor neuron, is gonna send the signals to the effector, which gonna be our muscle, which is going through contraction, or the muscle, which is going through the relaxation. For example, for the knee-jerk reflex, let's review all of the components. So we are going to stretch the length of the quadriceps femoris muscle, and then the stretch receptors, which also known as muscle spindle receptors within the quadriceps femoris muscle, is going to send the signal to the spinal cord, which means our controller is spinal cord. Okay, and then spinal cord through the motor neurons, through the efferent pathway, is going to send the signal to our muscles, the quadriceps femoris muscle, and also the hamstring muscles. So the response would be the quadriceps femoris muscle contraction, which it made our knee to extend, so knee extension, and then the hamstring muscle to relax. Next, we have triceps stretch reflex, which again, we have a receptor found within the muscle, and then the controller would be the spinal cord. The effector would be the triceps muscle itself. So triceps muscle to contract, and then we can experience extension of, so the response would be extension of elbow, by which gonna happen by contraction of triceps and relaxation of biceps. Next, we have pupillary light reflex, which we are going to shine the light through the eye. The sensors would be found in a retina of the eye, which are the photoreceptors. They are so sensitive about the light, so they're gonna send the signals through the optic nerve, cranial nerve number 2, to the brain. So our cranial nerve or optic nerve would be the sensory nerve working in the pupillary light reflex, and then the efferent pathway would be the third cranial nerve. So the sensors would be found in the retina of the eye. The afferent pathway would be the cranial nerve number 2, which gonna send the signals about the vision, about the light to the brain, and then the afferent pathway would be the motor neuron, which here we have the cranial nerve number 3, which is gonna send the signals, the motor signals, to the muscles of eye. So then the response would be pupil constriction. Lesion in the right cranial nerve number 3. What gonna happen for the pupillary light reflex result? One of your friend has a lesion in the right cranial nerve number 3, which is the motor neuron, which is the efferent pathway, which means it's the nerve or neuron which gonna send the signals from brain back to the eye. Then it's gonna have the response this way. So that is our cranial nerve number 3. Let's see what's gonna happen if the cranial nerve number 3 has been damaged on the right side. So here we have, if you shine the light to the right side, so the optic nerve of the right side is working perfectly, right? So it's gonna send all the signals to the right side of the brain. It's okay. But the way that the cranial nerve number 3 of the right side should send the signals back to the eye is not working. So it means the direct response for right pupil is abnormal. We're gonna send the signals, but then the motor signals cannot get back to the eye to get the response. So then we cannot see any response back. So it's abnormal. It means if you shine the light, you cannot see the constriction of the pupil for the right side because cranial nerve number 3 is not working. And how about the consensual response? You're going to send the signal to both sides, right and left, right? Okay, but the right side, the cranial nerve number 3 is working perfectly. So then we can see the constriction of the pupil on the left side. So that would be normal. And then how about the left side? If you shine the light to the left side, again, the cranial nerve number 2 is going to send all the signals to the left side of the brain, and then cranial nerve number 3 on the left side is working perfectly and send the signals back to the eye. And then we would have the normal result. What about the consensual response to at the right pupil? So then if you shine the light to the left side, then through the cranial nerve number 2, we can send the signals to the right brain with no problem. But the way that cranial nerve number 3 should get back to the eye is not working. So it means we're gonna get the abnormal result. Now, what about our left cranial nerve number 2 has been damaged? Okay, so here again, you can see when we are going to shine the light to the left side, okay, because the cranial nerve number 2 on the left side has been damaged. So the signals cannot reach to the brain. It cannot reach to the left side and right side. So it means when this light can, or this information cannot get to the brain, so there is no information here that the brain is receiving. So it means there is no like response back. So then direct response would be abnormal. What about consensual? The same, because there is no way that the brain can be aware what's happening in our eye, right? Because the cranial nerve number 2 is not working. So both direct response and consensual response on the left side would be abnormal. But what about the right side? So because the right side, the optic nerve is working perfectly, so it's gonna send the information to the right side and left side of the brain. So then we can get response back from the oculomotor number or cranial nerve number 3. So we would have the normal direct response, but for the consensual, because again, this cranial nerve number 2 does not have any problem to send these sensory signals to the left brain, right? So it means the left side of the brain is going to receive all of this information, and through the cranial nerve number 3 is gonna give the result. So then we're gonna see the normal consensual response. It seems confusing, but if you know the way that the cranial nerve number 2, the sensory neuron, cranial nerve number 3, the motor neuron is working, it's going to be so much easy. Thank you so much for listening, and good luck to you.